And as was noted, the pathogenicity of xenotropic MLV for an obvious reason, and that is this virus does not infect mice, has not been studied in mice, but closely related viruses have been very important pathogenic models in virology for cancer, for immunodeficiency disease, and for a variety of neurological diseases. So the potential pathogenicity of this virus in a host that it can infect should not be underestimated. So xenotropic MLV is inherited as an endogenous provirus, meaning it's in the germline, about 10 to 20 copies in all inbred mice, and in a much greater copy number actually in some wild mice. Most of the proviruses that are inherited this way are defective. They won't give rise to complete virus, but some of these are in fact capable of replicating. There's one called BXV1, which is intact and infectious. If you pull it out of the mouse DNA, you can get infectious virus out of it. And these viruses can infect virtually all mammals, and I think even some birds, except for some species of moose. And what's happened in these mice is that after the virus became endogenous, became integrated into the germline of the mouse lost the receptor for it, probably by selective pressure that was put on by the virus itself. So, as I said, these are not directly pathogenic in mice, but if you isolate pathogenic viruses, some pathogenic cancer-causing viruses from mice, they will have a sequence, a very important sequence, which is the long terminal repeat, which contains all of the signals for gene activation, for directing transcription of this, and for turning on oncogenes, which causes the cancer. And that LTR comes from the xenotropic virus, and it's very similar in sequence. Not quite identical, there are a few little differences, but it's very similar in sequence to the virus in XMRV. Another thing, and one of the big worries in this kind of study over the years is that these viruses are actually common in contaminants of human cell lines, because they infect cell lines like these prostate cancer cell lines very nicely, and these have often been isolated by passage through nude mice. And the nude mice have this infectious provirus, and this virus can then infect the tumor, and there are repeated reports over and over again of human tumor cell lines that have picked up these viruses in this way. And so when one first sees this, a major concern. The phylogenetic relationship, in fact, as Dr. Peterson pointed out, argues against that kind of, very strongly argues against that kind of contamination from laboratory mice. I already made this last point. Related murine leukemia viruses can cause a wide variety of diseases. This is a little cartoon that I published in the perspective that I wrote that accompanied the Judy Mikovits's paper describing the virus in CFS, and simply showing that the virus came originally from an exogenous virus, or an infectious virus infecting mice or infecting mouse ancestors, that then happened to infect the germline, so it became fixed as a fossil in the germline. Subsequent to that, the receptor got mutated, so the virus can still infect us, but it can't infect mice because they don't have the receptor, and then somehow it got transmitted to humans. The clusters and the very close relationship imply that this transmission may have occurred quite recently. And in fact, we don't know, but it's not impossible that it happens all the time. That there is some mouse out in the wild that has this as an endogenous virus, and my labs are actually we've just begun to look to see if that's true, and that the virus can be both transmitted from mouse to human and from human to human. We just don't. It just all remains to be seen, but it's, I think, a very important area of study to figure out where this virus is coming from, when it got into humans, and how, of course, it's being transmitted among people. On the right, I show the same kind of phylogenetic relationship, and just want to point out again the very striking similarity of the viruses isolated from both chronic fatigue and prostate cancer. There's no obvious division of viruses that are isolated from one disease or the others. The two most distant viruses on there are actually both from prostate cancer. And what is really a very close relationship to these endogenous proviruses and these infectious xenotropic viruses isolated from mice, and then the more distant relationship to other well-known mouse viruses. If we were to put HIV on there, I think you'd have to go all the way over to that wall and come back again to get the relationship. These viruses are very, very distant. They're all retroviruses, but it's very hard to even see the relationship when you try to line the sequences up. You just have to look in certain very, very specific areas. So it's important to keep in mind this is a very different group of viruses, a very widespread group of viruses, particularly in mammals, and even a few bird isolates. But never before, except in the case of some gene therapy applications, never before have these viruses been known to infect humans. And there was some effort actually put into looking at that particular issue in the case of feline leukemia virus back in the 70s when this virus was first discovered. There was a fair amount of concern that the cats might be spreading this virus into their owners. And this is actually my last slide, and I just want to emphasize what we don't know about this virus. This is very important. It's very, very early days in this. We've got one paper on associated with chronic fatigue syndrome, 2 papers on the association with prostate cancer, which don't completely agree with one another. And the paper, in my opinion, is as good as you get for the first paper on the subject, but it's still the first paper. There's much that needs to be done. And the first is, of course, to establish what the real role of this virus is in this disease. And this is not easy. It's not necessarily easy to establish causality. It took quite a while with HIV, despite the real smoking guns that you had. It's fairly easy to establish in some kinds of cancers because you can actually see integration site specificity that would point to it. But in other case of other diseases, whether the virus is the cause of the disease, a passenger, or just a geographic coincidence of infection with the disease, all of this remains to be established. Obviously, we're going to be NCI in particular is really mounting a big effort intramurally, at least, to develop this. We don't know the incidence and prevalence in the human population. I want to emphasize that in order to do these studies, it's very important that we have standardized, uniform, well-validated, and reliable assays. This is really the critical issue here. Immunological assays are particularly difficult in this case. Even with HIV, the well-established HIV assays still have a certain false positive rate. And so it's really critical that we know exactly what the assay is, that it's been well validated with well-standardized specimens, and everybody is on the same page, ideally using the same assay for these studies. And this is, I think, this is one of the things that we're actually working on in Frederick quite intently right now, is to try to develop some assays that will fill us bill. Other people are as well. We may win out, they may win out, but whatever happens, before we can really get a real handle on this, these assays are critically important. We need to know the distribution in the human population, of course. Are there clusters? Are there possible clusters of infection, or is it just generally widespread? We have no idea what the mode of transmission is, despite some things you may have read. In my opinion, we just don't know. It's the ability to easily isolate the virus from blood cells and from blood certainly implies that transmission could be bloodborne. Beyond that, I haven't a guess as to whether it's being transmitted by bodily fluids, it's probably not transmitted in aerosols. These viruses are not very transmissible that way in general, but we don't know that for sure for this virus. And we don't know the origin of this virus. The close relationship to these viruses means it almost has to have come from mice. I can't really visualize another scenario. But did that happen? Is that happening today? Does it happen all the time? Is it like a hantavirus outbreak or something like that, where or did it happen once a long, long time ago, and has been then transmitted at a low level in the human population? And of course, if it is causal for the disease, we don't know what the attack rate is going to be. We don't know what fraction of the people infected with this virus. Everybody infected with HIV eventually will die. Almost everybody, 99-point-something%, will eventually die of AIDS if untreated. However, everybody infected with another human retrovirus, HTLV1, only a small fraction actually get disease. So we don't know that for these viruses. We don't know. I mean, this list could go on and on. Actually, I want to say one thing about tests, though. it came to my attention and to my horror yesterday to see a website advertising a completely, as far as I can tell, undefined, unverified, unstandardized test for XMRV, and somebody, I think in South Carolina lab, wants $400 from you to test a drop of blood, and I think this is, well, I probably shouldn't say what I really think about this, but it's a very bad idea to have a test that we don't know what it is. We don't know how good. I'm sure the FDA and CDC people would echo this, and I would strongly urge anybody who's listening to this not to do this. I don't know we have no idea what the result would be.
Dr. Coffin, I want to